Welcome back to Dr. Liu's VCE Biology channel. Dr. Liu here. A lot of students tackling this 2025 exam question think that simply writing different footprint sizes is enough to score full marks for species variation. Wake up! If you only mention size, the VCE markers will assume you're just looking at a dad and a child from the exact same species, 0 marks. In today's video, we are skipping the fluff and diving straight into our logic ladder framework to dissect every single high-level trap in the 2025 VCE Biology Human evolution section. Whether you need to secure that precise mark for primate traits using opposable thumbs, or fully conquer that mandatory 2-mark question on the mtDNA molecular clock, this video is going to transform your common mistakes into guaranteed exam points. Don't forget to like and subscribe, and let's get straight into the high-yield content. The correct answer is B, the presence of an opposable thumb. To solve this using the syllogism approach, we can break down why the other options fail as distinguishing characteristics for the primate order. A and C, tail and fur, these are general mammalian characteristics. While the lemur has them, so do dogs, cats, and rodents. Therefore, they do not uniquely classify an animal as a primate. D is side-facing eyes. This is actually incorrect for most primates. A key primate feature is forward-facing eyes, which allow for stereoscopic, 3D vision and depth perception, essential for life in the trees. B, opposable thumb. This is a defining primate trait. It allows for a power grip and precision manipulation of objects, which is clearly visible in the image as the lemur grips the wooden logs. When identifying primates in exam questions, look for these specific syllogistic markers. Grasping extremities, opposable thumbs, or big toes in non-human primates, and flat nails instead of claws. Visual system, forward-facing eyes, binocular vision, and a protective bony orbit around the eye. Brain complexity, a large brain-to-body mass ratio compared to other mammals. Social or life history, long gestation periods and extended parental care. Note for students: in VCE biology, the distinction between mammals, primates, hominoids, and hominins is a frequent source of distractor options. Remind them that a feature like fur is necessary for being a primate, since all primates are mammals, but it is not sufficient to define one. A team of researchers has uncovered dozens of fossilized footprints at an archaeological site in Kenya. The sediments around the footprints were dated to about 1.52 million years old. The researchers made 2 conclusions. Conclusion 1: the footprints are from hominin species. Conclusion 2: there are 2 separate hominin species rather than variation within the one species. Question: describe the evidence that would support each conclusion. 2 marks. Conclusion 1. Conclusion 2. For a stem like the one provided, we use a highlight and categorize strategy to filter the noise from the mechanically necessary data. 1. The case study filter. We should look for 3 specific anchors in any VCE biology stem: time, place, and taxon. Time, 1.52 million years ago. This is a massive hint. It places the footprints in the Pleistocene. We should mentally recall which hominins existed then: Homo erectus, Paranthropus boisei. This prevents them from accidentally mentioning Australopithecus afarensis, too old, or Homo neanderthalensis, too young. Place: Kenya. Confirms the out of Africa context and supports the presence of multiple contemporaneous species. Evidence type: trace fossils. Footprints are trace fossils, not body fossils. This means the answer must relate to locomotion and foot anatomy, not brain size or jaw shape. 2. Parsing the conclusions. The stem provides two specific conclusions. We must treat these as the goal. Conclusion 1. Hominin species. The key info. The word, hominin. The translation. What makes a hominin unique compared to other hominoids, apes? The answer requirement. The student must provide a feature of obligate bipedalism. If they list a general primate feature, like having 5 toes, they will not get the mark because it doesn't prove the individual was a hominin. Conclusion 2. Two separate species versus variation. The key info. Rather than variation within the one species. The translation. How do we know this isn't just a big dad and a small child of the same species? The answer requirement. The student must find a structural or morphological difference. Size is a trap variable. 3. The mark allocation logic. Since the question is worth 2 marks and asks to describe evidence for each conclusion, the math is simple. 1 mark equals evidence for bipedalism, conclusion 1. 1 mark equals evidence for interspecific variation, conclusion 2. Conclusion 1. The footprints are from hominin species. To support this, you must identify features that prove the individuals were obligate bipeds. Non-opposable big toe. Unlike other primates, hominoids, the big toe in hominins is in line, adducted, with the other toes to provide a push-off point for walking. Arched foot. The presence of a longitudinal arch acts as a spring, absorbing shock and transferring weight during a bipedal stride. Pronounced heel bone. A deep, heavy heel strike indicates the foot was bearing the full weight of the body in a vertical stance. The presence of a non-opposable big toe in line with the other digits provides evidence of bipedalism, which is a defining characteristic of a hominin. Conclusion 2. There are 2 separate hominin species. The evidence here must point to structural differences that are too significant to be explained by age or gender within a single group. Distinct morphological differences. If one set of prints shows a significant difference in the angle of the big toe, or a vastly different arch height compared to the other, it suggests different evolutionary adaptations. Weight distribution patterns. Different pressure paths, how weight transfers from the heel to the ball of the foot, can indicate different gait mechanics specific to different species. Key distinction for students. Simply stating the footprints are different sizes is insufficient for a mark. Variation in size could easily represent a child versus an adult, or sexual dimorphism, male versus female, within the same species. You must emphasize anatomical shape or structural proportions. The footprints exhibit structural differences in arch height or toe alignment that exceed the typical morphological variation found within a single species. Use the following information to answer Part B below, and Part C on page 41. One species of hominins, Homo neanderthalensis, lived in Europe and West Asia for hundreds of thousands of years before disappearing about 40,000 years ago. Modern-day non-African Homo sapiens have inherited 1 to 2% of their DNA from H. neanderthalensis. Evidence for interbreeding between H. sapiens and H. neanderthalensis has been obtained from 2 recent studies, and is summarized in the table below. Each study analyzed the genomes belonging to H. sapiens individuals of different ages. B. Give one reason for the difference in the conclusion made in each study. Justify your response using information from the table above. To help students tackle data-heavy questions like this, we should break the analysis into two distinct phases: the command, the question, and the evidence, the table. 1. Analyzing the question, the command. Students often rush into the data without knowing what they are looking for. We can teach them to deconstruct the prompt. Give one reason. This tells the student they only need to identify one variable that changed between the two studies, e.g., sample size or time period. For the difference in the conclusion. This identifies the problem. The student must explain why the dates in the conclusion column don't match. Justify your response using information from the table. This is the most critical instruction. In VCE, justify means you must quote specific data points, numbers or dates, to prove your reason. If they don't use the numbers, they can only get 1/2 marks. 2. Analyzing the table, the evidence. Teach students to look for the delta, the change, across the rows. They should scan column by column to see where the studies diverge. Study 1 is a snapshot. With only 7 ancient genomes, the researchers were looking through a very narrow keyhole. Because the data was limited to a single point in time, they could only confirm a small 4,000-year window of interbreeding. Study 2 is a broad timeline. By increasing the sample size to 334 genomes, and including present-day individuals, the researchers opened the lens wider. This extra data allowed them to detect earlier and later genetic traces, revealing a much larger 7,000-year window that Study 1 simply didn't have the resolution to see. More data, higher N, and a wider time range usually lead to a more comprehensive and accurate biological conclusion. Here is a concise model answer that hits both marks. Study 2 is likely more accurate because it used a much larger sample size of 334 genomes compared to only 7 genomes in Study 1, allowing for a broader range of genetic data to be analyzed. Marking trap to avoid. Many students will simply say the conclusions are different. Remind them that the question asks why they are different. A justification in VCE biology must include the specific numbers or categories from the provided text to earn the second mark. Study 2 also compared the percentage of Neanderthal DNA in the 59 ancient human individuals to the 275 present-day individuals. The diagram below summarizes the findings. Account for the change in the percentage of Neanderthal DNA found in ancient human individuals compared to present-day individuals. 2 marks. To tackle this question, we need to understand that they are being asked to explain a process over time. Here is a breakdown of how to analyze the prompt and the visual data. 1. Analyzing the question, the command. Account for. This is a high-level command. It doesn't just mean describe what you see, it means explain the biological mechanism that caused this change. The change in the percentage. This tells the student to look for a mathematical or proportional trend. 2 marks. This signals a 2-part requirement. Identify that the percentage has decreased. Provide a biological reason, e.g., selection or dilution. 2. Analyzing the data, the visual evidence. We look at the key first, then the timeline. The black bars, Neanderthal DNA. Notice the physical length and number of bars. Top, greater than 40,000 Ya. The bars are long and frequent. This represents a genome shortly after interbreeding occurred. Bottom, present day. The bars are tiny fragments. This visual change is what they must explain. The y-axis, time, the arrow points downward. This indicates that as we move from the top of the diagram to the bottom, we are moving through thousands of generations of inheritance. 3. The biological mechanism. The change is a reduction in Neanderthal DNA. Students should focus on two main reasons. Genetic dilution, interbreeding. After the initial interbreeding events, Homo sapiens continued to breed primarily with other Homo sapiens who had little to no Neanderthal DNA. Over time, the Neanderthal signal was diluted in the larger gene pool. Natural selection. Some Neanderthal alleles may have been deleterious, harmful, or less advantageous in the environments humans moved into. These individuals were less likely to survive and pass on those specific genes, leading to their removal from the population over time. Concise model answer. The percentage of Neanderthal DNA has decreased in present-day humans. Because, over thousands of years, natural selection may have filtered out deleterious Neanderthal alleles, and continued interbreeding within the larger Homo sapiens population has diluted the original Neanderthal genetic contribution. Fossils have been used to indicate where, and at what time certain hominin species lived in the world. mtDNA has been used to provide evidence of how hominin species may have migrated across the world. Outline 2 benefits of using mtDNA to help determine pathways hominin species may have taken. 2 marks. For this 2-mark question, the examiners are looking for the unique properties of mitochondrial DNA, mtDNA, that make it a superior molecular clock compared to nuclear DNA for tracing ancestry. 1. Analyzing the question, the command. Outline 2 benefits. This requires 2 distinct biological advantages of mtDNA. To help determine pathways. The answer must link the property of mtDNA back to how it helps track migration. The trap. The official comments warn against describing how migration happens. Students must stick to the benefits of the tool itself. 2. The logic. Why mtDNA? Unlike nuclear DNA, which is a scrambled mix of both parents, mtDNA provides a clear, linear signal. Maternal inheritance. mtDNA is passed exclusively from mother to offspring without recombination. This allows scientists to trace an unbroken lineage back through generations. High mutation rate. mtDNA mutations occur at a relatively fast and steady rate. These accumulated differences act as a molecular clock to estimate when groups diverged. High abundance. There are hundreds of copies of mtDNA per cell versus only 2 for nuclear DNA, making it much easier to extract and sequence from ancient fossil remains. 3. Concise model answer, 2 marks. Maternal inheritance. mtDNA is passed down the maternal line without recombination, providing an unbroken lineage that makes it easier to track ancestral movement over time. High mutation rate. mtDNA has a known and relatively high mutation rate, which allows researchers to use it as a molecular clock to determine the timing of divergence between different populations. Primate classification, classified uniquely by opposable thumbs for precision grip. General traits like fur or tails are shared mammalian features, not exclusive primate markers. Hominin bipedalism, confirmed via trace fossil footprints showing a non-opposable big toe, longitudinal arch, and pronounced heel. Structural geometry, e.g., arch alignment, not footprint size, distinguishes separate species. Genomic interbreeding, larger sample sizes, n equals 334 versus 7, provide higher statistical accuracy, expanding the known Neanderthal human interbreeding window. DNA trends and mechanisms, nuclear DNA, Neanderthal DNA decreased over time due to natural selection against deleterious alleles and genetic dilution from continued H. sapiens interbreeding. Mitochondrial DNA, mtDNA optimal for tracing migration pathways due to maternal inheritance, no recombination, a steady mutation rate, molecular clock, and high abundance in ancient fossils. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.